TDP-43 is one of the most influential and most destructive proteins in neurodegenerative disease. When it becomes problematic, it forms toxic deposits inside neurons, and these deposits show up in nearly every case of ALS. The problem is that healthy TDP-43 is absolutely essential. Neurons rely on it to regulate RNA and maintain their day-to-day function. So researchers have been stuck in a difficult position. How do you target a protein that is deadly in one form, but necessary in another? A new approach from Mindwalk Holdings may finally offer a way forward. Their strategy focuses on what happens when TDP-43 changes into its disease-driving form. In its normal shape, the protein keeps certain regions hidden. But when the protein slips into the distorted shape, it unfolds and exposes a segment that is normally buried. Mindwalk's team used advanced computational tools to design molecules that recognize only this disease-specific shape, leaving the healthy version untouched. They built two complementary AI-driven systems that work together. One analyzes massive biological datasets to identify patterns in how proteins change as they become diseased. The other uses those insights to engineer therapeutic candidates that fit those changes with remarkable precision. Instead of screening thousands of molecules and hoping one works, the process starts with the exact structural features that define the distorted protein. Identifying the right target is only part of the challenge. TDP-43 does its damage inside the cell, far beyond the reach of traditional antibodies. To get around this, Mindwalk develops specialized antibodies that work inside neurons, often called Intrabodies. Because regular antibodies can't enter cells on their own, the Intrabodies are delivered using gene therapy tools that place the antibody's genetic blueprint directly into the neuron. Once it's there, the cell begins producing the therapeutic molecule itself, giving it an internal defense system against problematic TDP-43. Early lab studies show encouraging results. These Intrabodies help the cell clear out misfolded TDP-43, reduce the spread of toxic protein seeds, and interrupt the chain reaction that allows misfolded proteins to corrupt their healthy neighbors. By breaking this cycle, the approach aims to slow or prevent the progressive buildup of TDP-43 pathology. Distorted TDP-43 is also found in frontotemporal dementia and appears in subsets of Alzheimer's disease. Because this strategy targets the misfolded state of the protein, not a specific diagnosis, it has the potential to serve as a foundation for treating other neurodegenerative conditions that share this common pathology.